Looks like you got it.
Yep, we're preparing to live stream.
Okay, cool. I can start?
Yep.
Awesome. Hey everyone, we're just going to wait another couple of minutes just so everybody can come in. So just in the meantime, please mute your mics, excuse me. Okay, so I guess we can get started. Just like the other lectures, just keep your mics muted, please. If you have any questions, we can adjust them at the end of each section, or you can type them in the chat box. Janelle is also on, so she can answer any questions as well. So with that being said, let's jump in. Okay, so we're going to start with the basic structural and functional unit of the nervous system, which is the neuron. Neurons help your body's nervous system transmit information rapidly in the form of electrochemical impulses. They are highly specialized and they do not multiply, which is something interesting to keep in mind. Neurogenesis is the process of the generation of neurons, and it takes place mostly during embryonic development. But recent studies also show that it occurs in certain brain regions throughout adulthood, which is very interesting. While the endocrine system typically uses chemical signals in the blood to communicate with the target tissue, the nervous system uses its own network of different neurons throughout the body. Different types of glial cells also help neurons transmit information throughout the body. Glial cells are sort of like the support system for neurons. There are a couple of different types, and we'll definitely go over them. They do differ a little bit between the central and peripheral nervous system. But a lot of this lecture is just going to be straight content. Obviously, the nervous system is huge and there's a lot to cover, so it's kind of hard to condense everything into 2 hours. So just keep in mind that this is a review. We're just touching upon some of the basic points, but you should definitely go more in depth with your independent studies. All right, so let's take a closer look at the multipolar neuron. So the neuron pictured to the left is called a multipolar neuron. It's the most common neuron in the central nervous system of humans, and it's probably the one that you're going to see the most. If they ever give you a diagram, like on the MCAT, and they ask you to examine it, you're most likely going to see this one. I'm sure you guys are all familiar from your undergrad studies and high school studies. So let's just go over the anatomy of the neuron. So the dendrites are over to the left, the little projections that are coming off of the cell body. Those receive chemical information from other neurons. That's the input. They receive input there, and then action potentials are generated, which we'll get into a little bit later, and it propagates down the axon. But first, it has to pass the soma, which is the cell body. So like I said before, just keep in mind that the neuron, even though it's so different from other cells in your body, it still is a cell. It's the basic form of life, so it carries on all the metabolism of every other cell in your body. They are eukaryotic cells, and the body contains almost all of the same structures as other eukaryotic cells in your body. The axon carries information in the form of electrical impulses away from the soma to the axon terminal. And the axon of a single neuron can actually get really long. I think I put it in the slide somewhere later, but the longest axon in the human body is actually, it's up to 1 meter long, which is about 3 feet for those on the standard system. But it actually innervates your sciatic nerve, and it goes from your lumbar to the bottom of your foot. So I thought that was really cool that a single cell was like 3 feet long. The myelin sheath is an insulating layer of tissues formed by the oligodendrocytes in the central nervous system and the Schwann cells in the peripheral nervous system. So these are two of the glial cells that I was talking about before. They're support cells. They don't transmit information like neurons do, but they are essential for the nervous system. They insulate the axon, and we're going to talk a little bit more about them later. The axon terminal, which is also known as the synaptic terminal, it's known as the synaptic bouton. There's a bunch of different names. You should be familiar with all of them. I'm sure you've seen most of them. And what that does is it converts the electrical impulses received by the axon into chemical signals. So that releases the neurotransmitters into the synaptic cleft. It diffuses across the cleft, and then it touches the postsynaptic neuron. And I did, right at the bottom. The longest axon found in a human neuron is about 1 meter long, about 3.2 feet. I'm going to try to keep a pretty quick pace. So I'll try to stop at the end of each section if you have any questions. Does anybody have any questions so far? I know I'm talking kind of quickly. I just want to make sure I get through everything today. No? Okay. Remember, you can use the chat box. If I ever stop like this, you can also unmute your mic if you want to speak, if that's easier for you as well. Okay, so the 4 types of human neurons that you may encounter on the MCAT, the neurons vary in structure, but all neurons share the same general function. They transmit electrical signals throughout the body. The most common neuron that's found in the human nervous system, like we just spoke about, is the multipolar neuron. But if you take a look at these over here, we have unipolar, which just has a cell body with no dendrites coming out of it. A bipolar neuron, you can see the cell bodies in the middle, and it has 2 axons. They're myelinated. The pseudo-unipolar one has a cell body that's coming off of the axon, and the multipolar one is the one that we've already studied with dendrites coming out of the cell body. So you can see how they have similar structure. It's not really super necessary that you understand what each individual one does, but I just wanted to put it on this slide for a reference point. You should at least be familiar with the fact that there are different neurons and they do serve different purposes. But like I said, multipolar neurons are the ones that you're going to be seeing mostly. Okay, so we're going to do our first review question. I'll give you guys like a minute and a half to answer this. So which of the following structures would be missing from the soma of a human multipolar neuron? So think about what we just talked about, some properties of neurons compared to other cells. Even if you're not familiar with specifically what's in the cell body, you can definitely figure this one out. So I'm going to open the polls. Janelle, I think you have to open the poll.
Oh, okay. Let me do that. Is it up? Is it?
I don't see it.
It says it's in progress.
Okay.
Oh wait, someone answered. Okay.
They can see it.
Manure
Okay.
can see it. Okay. Yeah.
Okay, great.
You can't see it at all?
Nope.
Hmm.
Okay, take a minute. Okay, take another 10 seconds or so, get your answers in, and then we'll go over it. Okay. And we can end the poll and share the results.
Okay, do you see the shared results?
Nope.
No. Okay.
That's not too
Okay.
bad.
Yeah, it's not too bad. I'll just share the results with you then. So 40% said D. 20% said choice B, and 20% chose I have no idea. And then 10% chose C and 10% chose A.
Okay, so a decent spread. 40% of you that chose D, you are correct. So when you're thinking about neurons, you have to understand a couple of things. One is that they are a cell, just like any other cell in your body. They have to undergo metabolism, and they're alive. It's the basic unit of life, a cell. So it definitely has mitochondria because it needs to perform metabolism. It needs energy because, yeah. It has a nucleus, so the nuclear pores are also going to be there for that selective permeability. And C, it also does make proteins. The only thing that it should not have are D, centrioles. And the reason being is that they do not multiply. So they don't undergo mitosis, and centrioles are in the cell during mitosis, and they help mitosis. They form the spindle fibers to pull the two multiplying cells apart. So just based on the fact that neurons don't multiply, the answer is D. Is that clear to anyone? Does anyone have any questions? You can unmute. Great. Okay. So the next topic is going to be action potentials, in saltatory conduction. So I put a little graphic up there. I'll have a couple of more on the screen. Let's see. We'll talk about saltatory conduction in a couple of slides, but I definitely think it's worth talking about the resting membrane potential because it does come up on the MCAT pretty often in my experience. So the resting membrane potential, that should say, for a human neuron is about -70 millivolts. So what that means is that when you're looking at the inside of a cell and the outside of a cell across the membrane, the difference between the two charges on the inside and the outside is 70 millivolts. So the convention is that whatever that number is, you're talking about the inside of the cell. So if it's -70 millivolts, that means that the inside of the cell is 70 millivolts more negative than the outside, less positive than the outside. And how does the cell maintain this? How does the cell keep charge? Through ions. So there are two pretty important proteins that you should know about. It's maintained by the sodium potassium ATPase. It's also called the sodium potassium pump. So it pumps 3 sodium ions out of the cell and 2 potassium ions into the cell for every ATP used. So this is an active process, as I'm sure you know, because we're pumping these ions against their concentration gradient, their electrochemical gradient, I should say. So sodium, there's a high concentration outside of the cell, a low concentration inside. So if we want to pump it from in to out, we have to use ATP. And it's the same thing with potassium. Potassium concentration is high inside the cell, it's low out. So it's an active process when we pump potassium. So the other protein is a potassium leak channel. These don't use ATP, it's passive diffusion. So what the leak channel is, it's basically just an open channel that allows the cell to be slightly more permeable to potassium. So it's just passive diffusion. Potassium is going to flow down its electrochemical gradient. And as we just said, since potassium concentration in a normal cell is higher on the inside, the potassium is going to flow out. So the two of these work together and they keep in equilibrium. So resting membrane potential for a polarized human neuron is -70 millivolts. So the neuron at rest is polarized. When the dendrites receive a signal, the membrane starts depolarizing. So depolarizing is always describing the first half of the action potential. We are at -70, which is a pretty polar value. It's kind of far from zero. So as it approaches zero, it is depolarizing, hence the name depolarizing. It also does pass zero and overshoot. It goes, I think, to about 35 or 40. Opal said, what concentration is high where? So sodium concentration on resting neuron cells are higher outside of the cell. Sodium is always outside of the cell. Potassium is always high inside of the cell. Those are two things that you should definitely commit to memory because it comes up in, sometimes it'll come up in other topics other than the nervous system. Was that your question, Opal?
Yes, it was. Thank you.
Great, no problem. Okay. Yeah, definitely. I can repeat that. So sodium ions are higher. They have a higher concentration on the outside of the cell, and potassium ions have a higher concentration on the inside of the cell when it's just a neuron that's at rest. And then the sodium potassium pump and the potassium leak channels, they work constitutively to keep this equilibrium. Yes, and Janelle put that in the chat so you guys can see it as well when I move to the next slide. Okay, so now we can get to the depolarization. So I put another graph of the action potential right there. At this point, I'm sure we've all seen this graph a lot. So input received by the dendrites initiates a slight repolarization. Summation, I also wanted to mention really quickly. It's the process by which a neuron adds all excitatory and inhibitory input to determine if the action potential threshold will be reached. So what I mean by that is that if you have a single nerve that's receiving input, most of the time there are going to be multiple sources for that input. So there could be hundreds of neurons that are leading up to this. So all that's saying is that it's going to add all of the excitatory or inhibitory input from all of those neurons. And if it reaches that threshold of 55 that you see right over there, then the action potential is triggered and it's an all or nothing response. If you look a little bit more to the right where you see those failed initiations, sometimes the neuron will receive input, but it's not enough to raise that potential. It's not enough to raise the resting membrane potential past the threshold. So the failed initiation, nothing happens. And then the sodium potassium pump and the potassium leak channels just gradually bring it back to equilibrium right by that -70 value. So not all input turns into an action potential, but any input that reaches the threshold is a 100% all or nothing reaction. So, like I just said, if the membrane potential reaches the 50-55 millivolt threshold, the cell undergoes complete depolarization. The subsequent rapid depolarization is due to the opening of voltage-gated sodium channels. So that's what happens that's why the -55 millivolts is significant, because at that point, that is the proper voltage level that the voltage-gated sodium ion channels open. So sodium ions flow into the cell down their gradient, and it increases the charge inside the neuron. So take a minute and kind of like intuit that, wrap your head around it, because it makes sense when you think about it logically and think about it while you're looking at the graph. So once you reach the -55, before the -55, it's just these little depolarizations that were started by the input. So once it reaches that 55, all of the cell's sodium gates swing open really fast, and there's a huge influx of positive ions. So we're at -70, and we're getting a huge influx of positive ions. So that's going to rapidly go up past 0, up to about 40. So the depolarization propagates as an impulse down the axon to the synaptic terminals, where it's converted into a chemical signal, and it's released into the synaptic cleft as neurotransmitters. So that's when the electrical signal is converted into chemical. And that's how neurons synapse, and that's how they interact with each other and communicate with each other. So what channels open during each stage? So for this stage, you just have to know, we're going to go over, I think in the next slide or two, we're also going to go over how the voltage-gated potassium channels work as well. But those are more important for the repolarization phase after the peak. So yeah, so they are important, but for this specific section, just know that once the threshold is hit, the sodium channels swing open, and they swing open very rapidly. So that causes a rapid influx of positive ions, and that's what triggers the action potential. And once they're opened, you can't close them unless we go through a process that I'm about to talk about. Okay. Is everyone clear with that? Okay, so we're going to do another review question. Janelle, if you can start the poll. Why do I always do that?
Okay, poll is up.
Hold on 1 second, guys.
Oh,
Technical difficulty. Give me 1 moment. This is great. Hold on. Okay, hold on 1 second. I'm just going to stop sharing my screen for a minute. Okay. Okay, let's get back to that. Okay, sorry about that. Let's start up again with the review question. So what best describes the behavior of sodium ions just after the neuron's resting potential reaches the threshold? So sodium diffuses into the cell only because of the charge gradient established. Sodium diffuses into the cell because of the charge gradient and concentration gradient established. Sodium diffuses out of the cell only because of the charge gradient, or sodium diffuses out of the cell because of the charge gradient and concentration gradient established across the membrane. So take a minute, let us know what you think, and Janelle will share the results as soon as we're done. All right, guys, take like another 10 seconds and we'll go over it. Okay. All right, Janelle, if you want to close the poll and share the results.
okay. So I shared it. 56% said B, 22% said A, and 22% also said, I have no idea.
Okay, well, the majority of you did get it right. So sodium diffuses into the cell because of the charge gradient and the concentration gradient established across the membrane. So like we were just talking about, sodium is higher on the outside. So when you're talking about ions, it's really important to keep in mind that when they travel across a membrane, they're pretty much paying attention to two things. So it's going to be the concentration of sodium, which would be the concentration gradient, and it would also just be like the net charge gradient. So they're positive. So those are two things that are actually working. That's pretty important to know, not just for the nervous system. It's like especially important to know when you're talking about the electron transport chain, for example. The protons, they diffuse across the membrane according to their electrochemical gradient. So it has to do with concentration and charge. And just for that reason alone, A, B, and D are incorrect. Does anybody need further clarification with that?
What's creating? What is creating both gradients? Why is that gradients?
Yeah. It's just the fact that the charge is being carried by an ion. So it's just how things work. So if you have a concentration gradient separate from an ion gradient, say it's water diffusing across a membrane. Water is a neutral molecule, but if it's higher on one side than it is on the other, it's going to, go from high to low, because that's just how they behave. And it's the same thing with an ion. Say, for example, we have an electron, or just some sort of hypothetical particle that carries a charge but doesn't carry a concentration. If there's a higher charge on one side and a lower charge on the other, it's going to move down the concentration gradient. So when you're talking about an ion, you have to consider both variables. So there is a concentration gradient established because there's more sodium on one side than there is on the other. So it's going to move to the side that has less sodium because it's moving from high to low. But on the other side of the token, it's also paying attention to the charge gradient. There are other ions that have charge. So there could be chloride that have negative charge ions over there. There are potassium ions that we're familiar with. So that all factors into the ion gradient, the charge gradient as well. So I just wanted you guys to be aware that there are multiple variables that come into play when we're talking about diffusing across a gradient, especially if it's with ions like sodium and potassium. Okay.
Thank you.
No problem. Does anybody else have any questions? All right, fantastic. Then we'll move on. Samantha said, what are those factors? The factors, are you asking the factors that contribute to something moving across the gradient? Yeah, so basically just charge and concentration. They're going to move from high to low, and they're going to move from high concentration to low concentration, but at the same time, they're moving from high charge to low charge, positive to negative. So I just wanted you guys to be aware that there are multiple variables that come into play with that. But I mean, in terms of this, it's moving one way. The sodium is moving into the cell at this point in time.
Question. Yesterday you went over like the Nernst equation. Is that question how they might ask us about these concentration gradient as far as Nernst? Maybe?
So honestly, I did not really spend a lot of time studying the Nernst equation, just because I saw it on a couple of study guides, but the only time I've ever seen the Nernst equation on the MCAT personally is when they give it to you and they ask you to analyze the formula of it. So I'm not super familiar with it. But I mean, just in terms of the nervous system, I don't think they will ever ask you anything about the Nernst equation. They probably really won't even ask you much about gradients. They're just going to be talking about it in the context of the resting membrane potential. So I wouldn't worry too much about the Nernst equation. I would maybe just familiarize yourself about what it is, because I want to say most of the time, if it appears on the MCAT, they will give it to you. And if any other tutor thinks that's incorrect, please correct me, but that's just what I've noticed in my experience. Sometimes they can use the Nernst equation with the nervous system, but they would explain it or at least give it, right? Because it's a pretty complex equation.
Okay.
I personally don't really think it's worth memorizing.
Okay.
Okay. Okay, so now we're getting to the second half, repolarization. So when the membrane potential reaches about positive 35 to 40, I say that because it says 40 on this graph. Sometimes it says 35, but it's around that same general value. So the voltage-gated sodium channels become inactivated immediately, and they stop further depolarization. So as the membrane potential approaches this positive value, the voltage-gated potassium channels start to slowly open. So as you're getting closer to that top peak, the voltage-gated potassium channels are going to slowly open. And once you get to the top and the sodium channels close and the voltage-gated potassium channels open and they're completely open, this rapid repolarization happens. So again, if we think about it, intuitively, the potassium channels are opening and there is a high amount of potassium inside the cell. So it is going to rush out of the cell. So we're losing positive charges. Positive charges are leaving the cell. So that is going to make the cell more negative. It's going to bring the membrane potential down. So, and that's why we see this downstroke on the repolarization part of the action potential. Right. So the potassium channels remain open as the cell repolarizes back to its resting potential. So they're open this entire time. And the entire time on the downstroke, the voltage-gated sodium channels are inactivated, which is slightly different than closed, and we're going to talk about that in just a minute. The potassium channels remain open, the cell overshoots its membrane potential, and the potassium channels close at about -90 millivolts. So they actually don't close until you pass the membrane potential. So the potassium channels remain open until about -90 millivolts. They close, and then at that point, all of those channels, the voltage-gated potassium channels and sodium channels, they're not open for business. So we return back to the sodium potassium pump and the potassium leak channels. So they return from that -90 millivolts, they return the cell back to equilibrium right up to that 70 millivolts. So the membrane is unable to generate another action potential during this refractory period until the sodium potassium pump and potassium leak channels restore the resting membrane potential, and the voltage-gated sodium channels switch from inactive to closed. So when you're talking about voltage-gated ion channels, there are basically three forms that they can take. So there's open, closed, and inactive. When they are open, obviously is the easiest. They're open. Ions are flowing down their concentration gradient freely. When they are closed, they are closed. Nothing is flowing through, but they are ready to open. So the difference between inactive and open is that when a voltage-gated ion channel is inactive, it is impossible to open it until it switches back to the closed phase. So this is why action potentials have refractory periods. When that voltage-gated sodium channel closes right at the top of the action potential, it is inactive for the entire downstroke and for a little bit of this overshoot while it's repolarizing back to the resting state. So somewhere along right after hyperpolarization, as it's climbing back up to the resting state, it switches from the voltage switches it from inactive to closed, and then it's able to start the process again. But another action potential cannot be generated until that voltage-gated ion channel switches from inactive to closed. So Keith said, are the potassium ions already inside of the cell? Yeah, they should be. Potassium ions, the concentration is always highest of potassium ions inside the cell. So this repolarization phase, the reason why that works so well is because there's so much potassium inside the cell. So as soon as that ion channel opens, the potassium ions are going to rush out, right? Just as Janelle said, they're going to rush out, which causes the repolarization and the return to the resting state. So it releases a bunch of potassium. It hyperpolarizes to about -90. And then the sodium potassium pumps and the potassium leak channels slowly help restore it back to the resting state. And the process can repeat. Okay, anyone have any other questions on that?
Where on the graph?
Do you have a question, Opal?
Is the refractive?
Right. So technically, I guess you can consider the refractory period anytime after the sodium channel opens up, because the real limiting factor on the end of the refractory period is the voltage-gated sodium channel needs to switch from inactive to closed, from the inactive phase to the closed phase. And that happens right, that lowest point, right where it says hyperpolarization, between that point and the resting state is when the voltage-gated sodium channels will assume the closed position and they're ready for another action potential. So technically, I mean, I didn't put it on there because I don't think it's super important, but I guess it is worth mentioning. There are two types of refractory periods. There's the relative refractory period and the absolute refractory period. So the absolute refractory period is the one that we're talking about, where it's impossible to generate an action potential because we don't have use of the voltage-gated sodium channels. But once that opens up, as I said, it opens up before we actually get to that -70. So technically, as long as that is able to swing open at the right threshold, we can generate another action potential before we get back to the resting state. It just takes more input because we're not starting from -70 to get to -55, where we could be starting at -80 or -78. So it's going to take a little more input to reach that threshold. Was that helpful? Was that confusing? So the absolute refractory period, it is impossible to generate another action potential just because the sodium-gated ion channels are inactive. There's a quick period where the sodium-gated ion channels become active again, but we are not yet at the -70 resting state. So that small period is the relative refractory period where it's possible to generate another action potential. It just takes a little bit more energy, a little bit more input. Okay. And now we'll talk a little bit about saltatory conduction. So saltatory comes from the word for jumping. If anybody speaks Italian, speaks Spanish, I'm sure that Latin root looks familiar. And it describes the way that the action potential propagates down the axon. So action potentials are generated at the nodes of Ranvier between these sections of the myelin sheath. So if you look down at this graphic, it almost looks like these little bubbles of myelin that are going down the axon with little arrows hopping at those little points that are not myelinated. So those little points that are not myelinated are the nodes of Ranvier. So basically, it's pretty self-explanatory. I mean, saltatory conduction, it's just the charge is jumping from node to node to node. It's just more efficient that way. It uses less ATP because we're not continuously going down the axon trying to repolarize and depolarize and repolarize every single point. So it just jumps from node to node, and it actually speeds up the process and it makes it more energy efficient. And it also insulates. It also insulates from any background noise, any unwanted input from surrounding neurons as well. So myelin is a mixture of proteins and phospholipids that insulate neurons and speeds up the transfer of action potentials down the axon. It's produced by the oligodendrocytes in the central nervous system and by Schwann cells in the peripheral nervous system. Does anybody have any questions about that? It's just the basic method of how neurons, pass information from the cell body to the synaptic terminal. Awesome. Okay, so we're going to do another review question. So a neuron at rest receives input from another neuron, which causes the resting membrane potential of the cell to increase by 10 millivolts. How does this immediately affect the neuron's voltage-gated sodium channels? So the sodium channels remain closed, the sodium channels open. A small percentage of the sodium channels will open, but most will remain closed. Or the sodium channels inactivate and cannot receive further input until the resting membrane potential is restored. This is a little more complicated of a question, but think about it, take a minute, give us your answer, and we'll go over it. Yeah, so we can review that. So take another 10 or 15 seconds, get your final answers in, and we'll review. All right, Janelle, if you want to end the poll and share the results, we'll go over it.
Okay, so we have 54% saying choice A, 38% saying choice C, and 8% saying choice D.
Okay. Well, the answer is choice A. So the question says that the potential of the cell membrane is going to increase by 10 millivolts. So if you recall, resting potential for a neuron is negative 70 millivolts. So this brings us to a potential of about negative 60. In order to trigger an action potential, we need to reach that 50 number, 50 millivolts. So this is not enough to reach that. So when an input, when a neuron receives input and it's not enough to reach the threshold, nothing happens at all. The only thing, it just, the membrane potential rises a little bit, and then the sodium, potassium pump, and potassium leak channels just slowly bring it back to resting potential, unless another input is applied and it reaches that threshold. But B, the sodium channels open. They don't open because we didn't reach the threshold. C, a small percentage of sodium channels will open, but most will remain closed. That doesn't happen just because, remember, action potentials are an all or nothing threshold, and every single sodium channel that's in the cell has that same voltage sensitivity. So it has to reach like about that negative 50 in order for all of the channels to swing open. So D, the sodium channels inactivate and cannot receive further input until the resting membrane potential is restored. That's not correct because we just have to be careful when we distinguish between inactive and closed. So it's only going to inactivate when the cell hyperpolarizes. So I'm sorry, when the cell depolarizes and passes zero and goes to a positive resting potential. So that's when we see the sodium channels inactivate, is when we're at that positive 35, positive 40. And then on the come down, once we overshoot that resting potential and we get to negative 90 on the repolarization phase, that's when the sodium channels switch from inactive to closed again, and the cycle can repeat. However, if we never reach that threshold and an action potential is never generated, the sodium channels don't inactivate. Nothing happens to them. They just remain closed. And they're still ready to go because nothing happened. Does that answer everyone's questions? Anybody have any other questions? Okay. Okay, so let's talk a little bit about synaptic transmission.
I thought you were going to go over.
What did you want to go over, Opal?
Remember the slide before, saltatory conduction?
Oh, sure. So this is being, hold on one second. It's just giving me some technical difficulty with these slides. Okay. Okay, so this is the slide you wanted to go over, right, Opal? Do you have any specific questions, or you just want me to review saltatory conduction as a whole?
Review it. It's just that there's questions in the exams that will talk about an unmyelinated neuron.
Yeah.
What happens? Oh, it's right there. So what's the deal with unmyelinated versus myelinated? Does it make the conduction faster or slower?
So myelin generally makes the conduction faster. I'm not too certain about like the structure, the function, or like the practicality of unmyelinated neurons. I haven't really studied them super in depth, but what you need to know about myelin is pretty much all you need to know is on this slide. It's just a mixture of proteins and phospholipids, and it insulates the neuron. So if you think about like a wire coming out of the back of your TV, there's copper, there's metal on the inside, but it's wrapped, it's insulated because it helps, it blocks out any interference from outside, and it helps the message get where it needs to be quickly. So you can kind of think of it that way. The only difference, of course, is that for a neuron, we have these little nodes of Ranvier, and it causes saltatory conduction. So you can think of it actually like a jumping action. So an action potential starts right over here, right by the body. Once that fires, it is enough to depolarize the membrane all through the myelin sheath, and then it goes to this one. then it goes to the next one, and then it goes to the next one, and then it goes to the next one. And that's just the mechanism of transmitting.
Is this what's going on in muscular dystrophy, where they're not able to respond? Is that
So that might be the case. I know that there are certain conditions where your motor functioning is affected because of a lack of myelin. So I'm not sure specifically if that is for muscular dystrophy, but that is definitely a consequence of unmyelinated axons. Yes.
Yeah, I think those are the questions I usually come across.
Yep. Yeah, they like to relate it to technical, obviously. No problem. Okay. Okay, so synaptic transmission. Just gave you a quick graphic over here. As you can see, there's postsynaptic and presynaptic neuron. Presynaptic is obviously first. It releases neurotransmitters, and they travel across the synaptic cleft, which is right over here, onto the receptors. And that's what I mean when I say input. I've been using the word input a lot. It's just any sort of signal that the postsynaptic neuron receives from anything before it. So in this case, it releases a neurotransmitter. It diffuses across the synaptic cleft. It attaches to these receptors. And sometimes yes, sometimes no. It's enough to depolarize the membrane enough to hit that threshold and trigger an action potential. So it's basically, nervous system transmission is basically just a series of that throughout your body. So there are two types of synaptic transmission, electrical and chemical. Electrical, it's usually just discussed very quickly and touched upon. It's used in cells that are attached to each other by gap junction. So these are not really neurons. Neurons don't really engage usually in electrical synapse. This is for like cardiac tissue, like smooth muscle cell. They're actually connected, and the cytoplasm of them are connected. So they can communicate very easily and very quickly. It allows muscle to contract uniformly. Right, it's mostly found in smooth muscle cell, cardiac muscle. Gap junctions and electrical synapses are also responsible for coordinating muscle contractions during childbirth, for example. The action potential can directly spread from one cell to the next because of the connection. So it's kind of similar to the electrical impulses of a neuron, but instead of when it gets to the end of the cell, it doesn't release neurotransmitters or release any sort of chemical. The electrical signal just goes right through those gap junctions. Gap junctions are just small openings that are connecting adjacent cells. So because the cytoplasm it's continuous, the ions can flow freely through the electrical synapses, and it makes for very highly coordinated movements and contractions. So the other one, which is more of our area of interest for this lesson, are chemical synapses, which we were just talking about. So those are found where the synaptic terminal meets its target cell. It could be an effector, which is like a muscle or some sort of organ, or it could just be another neuron, like we saw in the graphic before. So the two cells do not make direct contact in a chemical synapse, which is important to understand. The synaptic cleft is the little space between the two of them. So they can't touch, they're not touching, and the way they communicate is chemically. So that electrical signal is converted into a chemical signal, it crosses over the synaptic cleft, and then it is again turned into an electrical signal to travel down the second axon of the second neuron. The synaptic terminal releases the neurotransmitters via exocytosis. So what that means is that there are little vesicles inside the synaptic terminal that contain the neurotransmitter. These little vesicles, whenever an action potential reaches the end, they move closer toward the membrane. The membrane sort of associates with the vesicle, and then it sort of spits out the neurotransmitter, and it travels across the synaptic cleft, as we said, and it touches the postsynaptic neuron. So they initiate action potentials to continue the transmission of the information. And after the transmission, neurotransmitters are degraded. There are some enzymes that degrade the neurotransmitters while they're in the synaptic cleft. And then there's also a process known as reuptake, which is of importance in modern medicine, especially I'm sure some of you have heard of selective serotonin reuptake inhibitors. I see a question about that at least once on almost every MCAT that I've taken. They really like to ask that. And it's a pretty good way to gauge your knowledge about the nervous system, I think, and psychology. So reuptake is just exactly what it is. Taking the presynaptic neuron takes up any of the neurotransmitter, and it recycles them, and it can use them again. And the degrading of the neurotransmitters and reuptake is also what stops the action potential. So if these processes didn't happen, then there would just be neurotransmitter just chilling in the synaptic cleft, binding to the postsynaptic neuron and just like firing haywire. So the way that we stop that, the way that we stop the transmission, we degrade some neurotransmitters and some are taken up by the presynaptic neuron. So Keith said, what is a gap junction? A gap junction is just a small connection between adjacent cells. So say you have two cardiac cells, they're next to each other, they will have little pores that are connected to each other. And their cytoplasm it's continuous throughout them. So whenever one cytoplasm hyperpolarizes, if there's a change in the ion concentration, it's going to carry over to the next one. So yeah, like Janelle said, it allows direct cell-to-cell transfer of ions and small molecules. So it's super efficient, and it allows these cells to communicate almost instantaneously. Not instantaneously, because it does take, milliseconds. Yeah, through a channel. They're just like, think of them as little pores. So if you have two cells that are up next to each other, there are little holes that are connecting the cytoplasm of the two. And it's, like I said, they're confluent. So whenever one change happens, it very quickly changes the cell to the next of it. It's just another way. We use the word synapse, even though it's a little bit different than a chemical synapse, because they are touching, but it's just a way that the two cells communicate with each other. So like for cardiac tissue, when you send an electrical impulse, you want all of that tissue to contract at the same time. So if you use a chemical synapse, there's going to be a little bit of a delay. So electrical synapses do act a little bit quicker, and it allows for coordinated movement. Anybody have any other questions on that? Cool. Okay, so neurotransmitters. You should be familiar with the neurotransmitters and how they both oversupply and undersupply affects the human body. I don't know what I was going for with that one, but this to the left, it gives you all of the major neurotransmitters that you'll see. Acetylcholine, definitely dopamine, serotonin. I would definitely familiarize yourself with all of these, their basic functions. And then just maybe, yeah, like for the two lower ones, if they're inhibitory, if they're excitatory, usually GABA and glutamate, those are really the context in which they'll ask questions, is really just if it's inhibitory or excitatory. And I would definitely, I'm not going to spend too much time going over it, but this is really good for your own studying purposes. I've noticed that the MCAT really likes to test malfunction. When they talk about neurotransmitters, they love testing malfunction. So like I've seen a lot of questions where it asks, what happens when there's an oversupply of dopamine. So you should be, pretty familiar with, okay, if there's an oversupply of dopamine, that's linked to schizophrenia. Those are pretty common questions that I see on the MCAT. So the parasympathetic nervous system uses acetylcholine to transfer information across synapses. So keep that in mind. The parasympathetic nervous system, which we will talk about in a couple of slides, is a division of the autonomic nervous system. It uses acetylcholine basically exclusively as a neurotransmitter. And the sympathetic nervous system uses acetylcholine as well for preganglionic neurons and norepinephrine for postganglionic neurons. And if you need a little refresher on what pre and postganglionic neurons are, we're going to go over that a little bit as well. Does anybody have any questions about the neurotransmitters? Anything about the malfunctions? Anything about their functions? How you may be tested? Because the stuff that I put to the right, we're gonna review that now within the next couple of slides. So don't be freaked out if you see a couple of terms on this slide that you're not familiar with. Right. Yeah, so they're, like Janelle was saying, the second section, you will see the nervous system a lot, because the nervous system does pop up a lot, especially with psychology, when you're talking about cognition. Even when you're talking about clinical psychology, malfunctions of the nervous system are responsible for a lot of that stuff. So going over the nervous system is pretty important. It'll help you in multiple sections. It will help you in multiple sections of the MCAT. And we're going to do another review question. So like we were talking about before, SSRIs are a common treatment for major depression. Which statement best describes their mechanism of action? So just based on what we've been talking about, we spoke about reuptake before. These are selective serotonin reuptake inhibitors. So do they decrease the rate of degradation of serotonin in the synaptic cleft, therefore increasing free serotonin levels? I'm just going to let you guys read it. Take a couple of minutes, think about it, and we'll regroup in a few minutes and discuss. Okay, we'll take like 15 more seconds, get your answers in, and then we'll go over it. All right, Janelle, would you be so kind as to share the poll results?
Of course. Okay, so this one was quite a spread. 33% said A, 25% said B, 17% said D, 17% said I have no idea, and 8% said C.
Cool. Okay, so let's go over this. The correct answer is D. There was a lot to read and there was a lot of tricky wording, but I just wanted to, make these questions challenging so that we can review and we can really understand. So A, SSRIs decrease the rate of degradation of serotonin in the synaptic cleft, therefore increasing free serotonin levels. It's not that, just because that's not the mechanism of action for selective serotonin reuptake inhibitors. So, I mean, it does help if you have previous knowledge of how SSRIs work. Like I said, I see them a lot on the MCAT, so you definitely should know what they are, like the mechanism of action and what they're used to treat. But if you just look, it's a reuptake inhibitor. And we just talked about reuptake being the reabsorption of neurotransmitters from the synaptic cleft back into the presynaptic neuron. So serotonin reuptake inhibitors don't decrease the rate of degradation because they don't really interact with any of the enzymes that are degrading the serotonin. So SSRIs increase the rate at which the presynaptic neuron absorbs leftover serotonin from the synaptic cleft, increasing intracellular serotonin levels. That's actually the opposite of what SSRIs do. That's the opposite of their mechanism of action. So the thought process behind SSRIs is that if we actually keep serotonin out of the cells, out of the vesicles, and we keep it in the synaptic cleft, it's going to increase the firing. There's going to be more serotonin. There are higher extracellular serotonin levels. So that's actually the mechanism of action. That's the sort of train of thought the logic behind SSRIs. So B is actually opposite of what we're trying to do. C, the SSRIs bind reversibly to postsynaptic receptors to increase the concentration of free serotonin in the synaptic cleft. So C would hypothetically have the SSRIs kind of acting as an antagonist or like a competitive inhibitor, which is not the case either. They don't really do anything with the postsynaptic receptors. So yeah, like I said, they inhibit reuptake. So the SSRIs reduce the speed at which the presynaptic neuron reabsorbs serotonin. So reabsorbing serotonin is reuptake. Reducing the speed at which that happens is inhibition. So we are inhibiting reuptake. So it keeps the serotonin levels in the synaptic cleft higher for a longer period of time. And Anna, yes, well, it's very difficult when you're talking about pretty much any social science. It's hard to make cause and effect statements. So I think it would just be a more accurate statement to say that low serotonin levels are correlated to depression. But yes, when we're talking about low serotonin levels, we're talking about the serotonin levels that are extracellular, that are in the synaptic cleft. So Opal said, yep, Opal said if SSRIs are keeping serotonin in the cleft, there is constant stimulation firing, so depressed people are also excitable. Not necessarily excitable. I mean, that may be a side effect, but the takeaway is just basically what you said in the first part of that question, is that there's more stimulation and more firing of that pathway, just because the pathways that utilize serotonin as a neurotransmitter are linked to happier moods, higher moods, and it seems like, you know, at this point in time, the clinical information does support the fact that SSRIs have a significant effect on helping depression.
So they give depressed people SSRI drugs? Is that how they treat depression, by giving them?
Yes.
Okay.
Yeah. I mean, there are a bunch of different ways to treat. I mean, like if you go to a psychologist or a therapist, they'll be more like psychotherapy based. You can go to an MD psychiatrist, and yeah, they will often prescribe SSRIs. Yep. Cool. Okay. Any other questions before we move on? Definitely familiarize yourself with SSRIs. Okay. Awesome. All right, so let's get into the organization of the human nervous system. So I put a little graphic, slightly freaky graphic to the right, and it sort of separates the central nervous system, which is just the brain and spinal cord. That is pretty much the executive for all of your nervous functioning. And then the peripheral nervous system is everything else. So all of your sensory organs, any nerves that are coming off of your spinal cord, any nerves that are connected to your brain, that innervate your entire body, they receive input. Your sensation and perception, that's all captured by the peripheral nervous system. It's shuttled to the central nervous system and then back out to the peripheral nervous system. So we're going to talk a little bit about that. So here is a flow chart of the nervous systems. The central nervous system is off to the left. Obviously, there's nothing branching off of it because, as this says, it's mission control. There are no significant ways to further divide the central nervous system, but the peripheral nervous system, obviously, because it's much larger and it has a lot of functions, there are a lot of subcategories we can divide it into. So the first two being sensory and motor neurons. So your sensory neurons conduct signals from the receptors to the central nervous system. Receptors are, any sort of sensation input, like if you feel a touch. The sensory neurons are responsible for taking that input, bringing it right up to the central nervous system. The central nervous system will process it, probably send it to some lobe in your brain that's responsible for processing touch. It'll come back down, and that's how we feel the sensation. Those are also called afferent neurons. So the difference between afferent and efferent is that one is going towards the central nervous system and one is coming away from the central nervous system. So a trick that I use to remember that I was taught in undergrad is that afferent is arriving. They both start with A. So afferent is arriving and efferent is exiting. So AAEE. So you can keep in mind that sensory neurons, are afferent because they are arriving. Motor neurons are efferent because they are exiting. Anna, you said parasympathetic and GI is opposite, right? What do you mean? I know the parasympathetic response on the GI tract, there's a specific parasympathetic response that we'll talk about in a little bit. Is that what you're referring to? Increases activity? Yes, if that's what you're talking about, yes. For the most part, the parasympathetic response will increase GI activity, which we're going to get into in a couple of minutes. Keith, so the central nervous system is just your brain and your spinal cord. It is mission control, as this diagram says. It basically makes all the decisions for your nervous system. The peripheral nervous system is everything else. Anywhere we're getting input, anywhere in your body, your nervous system, the nerves, anywhere that's not your brain or spinal cord is your peripheral nervous system. So it's basically the system that brings information to and from the central nervous system. So the afferent neurons, as we just said, bring it to the central nervous system, they're processed, and then motor neurons or efferent neurons bring them away from the central nervous system. So those motor neurons, can be further divided into two more categories, the autonomic nervous system and the somatic nervous system. So the autonomic, obviously, as it says, is the involuntary response. So that's your sympathetic and your parasympathetic responses, fight or flight. It mobilizes bodily systems, parasympathetic rest and digest response, and conserves energy. So we often think of the sympathetic and parasympathetic response as a response to like an immediate stressor, which often is the case, and a lot of times you will see it in that context on the MCAT. But it's important to know that the sympathetic and parasympathetic systems also just mediate, your day-to-day nervous system activity. So I think I have a couple of examples on the next slide. And the somatic nervous system, I think we'll talk about a little bit as well, is just the voluntary movement control. So that's just your voluntary muscle. The skeletal muscular system is innervated by the somatic nervous system, that striated muscle that we control. Okay, so a little bit more on the sympathetic and parasympathetic response. The sympathetic response is the fight or flight response that is activated when an animal is exposed to a stressor. But obviously, like I said before, it is also still active when we're not in the presence of a stressor. But I would definitely try to know as many of these as possible. Once you figure out the pattern and you really have an understanding of the sympathetic and parasympathetic response and what their goals are, you can start to intuit this. So like if you're not like really keen on remembering things, just like bland memorization, you can figure it out. So like digestion slows, for example. We know in the fight or flight response, epinephrine is released. Digestion is going to slow because if you're in a life-threatening situation, it's not very efficient to divert blood flow and divert energy to digesting food when you have a 600-pound animal in front of you that's trying to kill you, for example. Same thing, urination is inhibited, bronchodilation occurs, your lungs open up, we're increasing oxygen intake because we're burning energy really fast because we need it in that situation. Sweating, vasoconstriction occurs. The vasoconstriction, your blood vessels get pretty small, that increases your heart rate, that increases your blood pressure. So we're getting more oxygen, more nutrients to the target tissue faster, and more of it in a small amount of time. Dilation of pupils occur. It allows you to see farther distance, the release of epinephrine. And one example of the sympathetic response not during a time of stressor is the sympathetic response is responsible for orgasm during sexual functioning. So on the other side of the coin, the parasympathetic response is the rest and digest state that an animal enters when no stressor is pregnant. So in the context of fight or flight, you can think of the parasympathetic response as the come down. Digestion starts to be active again because now we're trying to conserve our energy. We're trying to carry on our regular metabolic functions as efficiently as possible because we're not in the presence of a stressor anymore. So we don't need to divert our energy to the current situation. So again, everything else is intuitive once you understand it. So the digestion is active, which makes sense because it slowed when we needed to divert energy away from it. But now that we have the time, we're going to digest our food and get our nutrients. Urination may easily occur as well. Airways are constricted. That doesn't mean that they close up to the point where you can't breathe. It just means that they're not dilated to the point as they would be if epinephrine were present, as indicated by the sympathetic response. pupils are constricted, same thing. We're focused on energy conservation and metabolism. There's an increase in saliva production because, as Anna mentioned before, and as we just spoke about with digestion being active, saliva production is a part of digestion. So that's going to increase when we're resting and digesting. Vasodilation, our blood pressure goes down, our heart rate goes down. We don't need to spend so much ATP pumping all of that blood because we're not using as much. And again, I just wanted to put right at the bottom that just daily activities, sexual arousal as opposed from orgasm is actually mediated by the parasympathetic response. Well, as Anna so colorfully put, sometimes epinephrine and other hormones, cortisol is a stress hormone, for example, that can be released during the fight or flight response. So those sort of anxious responses that you will have, they're going to happen just because of the situation that you're in, and that's more of a psychological thing. But from a GI and a physiological standpoint, digestion does slow. So, I mean, if you had something, in the tank ready to go already, then that might exacerbate it. But the digestive system as a whole does slow, and blood flow is diverted away from it. Hope that kind of answers your question. And does anybody else have any questions on this before I move to the next slide? So Opal said, if vasoconstriction is occurring when I'm running away for help, how do I scream? Well, vasoconstriction is your blood vessels. So I don't think that they will constrict enough to make you not able to speak. So hopefully, if screaming is your plan of action and it works for you, hopefully you'll be able to scream. You guys got some interesting questions. Why, when some people are dying, they pee or go poop? I don't think that has anything to do with the sympathetic or parasympathetic response. I think it just has to do with like the body muscle contractions after death, rigor mortis, which I'm sure we're going to talk about in other lectures, how ATP plays a role in muscle contraction and why that happens after death. But A+ for being inquisitive. Okay. All right. Anything else before I move on? Cool. Okay. So I heard that you are stronger when you're in fight or flight. Yeah, some people do report that. I mean, it would make sense because epinephrine, we'll talk about this again in another lecture when we talk about metabolism and how your body regulates metabolism. Epinephrine is very good at mobilizing your glycogen storages, your glucose. It gives your body all that glucose that you need. So your muscles, they have as much energy as they're going to have whenever you have epinephrine circulating. So you're at your maximum strength pretty much. Okay, let me continue with this for time's sake, just because I want to get through. So the central nervous system is organized spatially. We won't spend too much time on this. I just wanted to add this graphic because I feel like I've seen it a lot, especially when I'm studying throughout like undergrad and even in high school. This graphic, known as a homunculus, demonstrates the fact that the cerebral cortex has specific regions that control specific functions. So that means exactly what I wrote in the title. Just keep in mind that the central nervous system is organized spatially. So there are specialized areas in the brain and specialized area in the spinal cord that handle specific neurological functioning and specific nervous functioning. So the central nervous system integrates information from the entire body to coordinate activity, like we said. And remember that the central nervous system is, again, made up of the brain and the spinal cord. Okay, so afferent neurons may synapse with interneurons in the central nervous system before relaying information back to the efferent neurons. So as we mentioned before, afferent means that they are arriving to the central nervous system. and efferent means that they are exiting from the central nervous system. So sometimes they synapse with each other, and sometimes they synapse with interneurons, which are just smaller neurons that can sort of alter the message. So like, for example, we sometimes talk about them in reflex arcs, where they'll sort of change an excitatory impulse to an inhibitory impulse. But that's really all I want to go in depth with in terms of interneurons right now, because I think it's just more important that we understand the role of afferent and efferent neurons. Okay, yeah, I mean, so when you have some free time, if you want to just stare at this diagram, sometimes it's trippy, but it's kind of interesting, and it's pretty useful. Okay, so moving on to the peripheral nervous system, we have sensory neurons that receive information and relay it through the dorsal root ganglion before relaying it to the central nervous system. These are known as afferent neurons. So like we said before a couple of times in this lecture, sensory neurons are afferent neurons. Afferent neurons are arriving neurons. So they are coming from your body to the central nervous system. And I used the phrase dorsal root ganglion. I said post-pre-ganglionic cells before. So ganglia, which is the plural of ganglion, are clusters of cell bodies. And on that note, nerves are bundles of axons traveling in the same direction. So for example, we talked about the sciatic nerve a little bit before and how it's just one single axon. The length of it is one single axon, but the actual nerve is a bundle of different neurons. So a nerve is a bundle of axons, and the ganglia are clusters of cell bodies. So these sensory neurons, these afferent neurons, have their ganglia close to the central nervous system, and we refer to that as the dorsal root ganglion. So the information goes there, and then that is where they synapse with the efferent neurons. So motor neurons carry information away from the central nervous system to target tissue. These are also known as efferent neurons. So just know that the dorsal root ganglia, they're located just outside of the spinal cord, and it's where the two different types, the afferent and efferent neurons, communicate with each other. So that's where they access the central nervous system, they're processed, and then they go back out. Okay, a little bit more on the autonomic nervous system. Efferent nerve pathways in the autonomic system and the autonomic nervous system consist of a preganglionic and a postganglionic neuron. It's pretty synonymous with presynaptic and postsynaptic neuron. It's just specifically talking about the ganglion. So sometimes neurons can synapse outside of a ganglion, but for all intents and purposes, it's the same thing here. Preganglionic neuron cell body is in the central nervous system. It synapses with the postganglionic neuron outside of the spinal column. So it's considered part of the central nervous system, but the actual synapse is just outside of the spinal column. So if you're familiar with like the planes of the body and the words we use to identify, like dorsal and ventral, for example, ventral, I believe, is stomach, like the front of the body, and dorsal is towards the back of the body. So that's where the name dorsal root ganglion comes from. It's dorsal to the spinal column. So that's where that name comes from, and that's where those neurons synapse. The postganglionic axon innervates the target tissue. The target tissue is also known as an effector, E-F-F-E-C-T-O-R. And that gives the expected response after the postganglionic neuron gets the action potential over to the effector. So the relative sizes of post and preganglionic neuron axons differ between the sympathetic and parasympathetic nervous systems. It might seem insignificant, but it's not a lot to memorize. I would definitely memorize these two bullet points just because I have seen them on the MCAT before. It's not like a huge deal to memorize them, so I think it's definitely worth your while. So the parasympathetic preganglionic axons are long, and their postganglionic axons are short. So we're talking about an entire system of two axons. The first one, the afferent axon for the parasympathetic nervous system, is longer, and then it's going to synapse closer to the effector. So that's after it synapses, that second axon only has to be shorter. And a way for you to remember that, which I've seen in a couple of different study materials, is paralong pre. So the parasympathetic has a long preganglionic axon. And then opposite, the sympathetic preganglionic axons are shorter because the ganglia are located closer to the central nervous system. So that preganglionic axon, that afferent neuron from the sympathetic nervous system is going to be a lot shorter. Again, because the ganglia are located closer to the central nervous system. So they're going to synapse, and then that postganglionic axon is going to be much longer because it needs to travel from the central nervous system and it needs to reach the effector, the target tissue. Is that clear? Do you guys understand that? Any questions on that? And I know a lot of this, there's so much terminology that's being thrown around because there's just so much material on the nervous system. So you just have to be very careful and just be very diligent with your independent studies. You can definitely use the slideshow, look back on the Zoom. I recommend, pausing, taking notes, definitely doing that, but use it to guide your independent studies because there may be some topics on here that we don't touch on that are worth your while. Reviewing. Okay. And we're going to do this one more question. I think this might be the last. So take a couple of minutes. A postganglionic sympathetic neuron is found to have a myelin deficiency, therefore preventing any action potential from reaching the target tissue. Which neurotransmitter is most likely prevented from being released from the postganglionic neuron? So this is just a basic comprehension question, but it's one of those you either know it or you don't. So take a minute. All right, guys, take about 15 more seconds, finish up, and then we're going to review it. Okay, and Janelle, would you like to share the results with us? With me?
Yep. I'm ending polling. Okay, so I just shared it. 50% said A, 17% said C, and I have no idea, and 8% said B and D.
Nice. Okay, so unfortunately, the answer is B. So let's just reread the question again. A postganglionic sympathetic neuron is found to have a myelin deficiency, therefore preventing any action potential from reaching the target tissue. Which neurotransmitter is most likely being prevented from being released from the postganglionic neuron? So we did mention quickly earlier in the lesson that the parasympathetic nervous system is mediated by the release of acetylcholine. So both postganglionic and preganglionic parasympathetic neurons use acetylcholine to communicate with each other. When we're talking about the sympathetic nervous system, the preganglionic neuron does use acetylcholine. So if we were talking about the preganglionic neuron, that would have been correct. However, most postganglionic neurons in the human body use the neurotransmitter norepinephrine. So if the action potential was not able to be generated on the postganglionic neuron and it wasn't able to communicate with the effector, with that target tissue, norepinephrine would not be released. it would be prevented from being released because no action potential would reach that synaptic terminal. No vesicles would exocytose, and there would be no norepinephrine released. So yeah, that was kind of an obscure question, but does anybody have any questions about that? Anything they want me to explain regarding that? So acetylcholine is used by the entire parasympathetic nervous system, and it is used by the preganglionic sympathetic. So if you want to, a good way to think about it is maybe if you divide it into 4 sections. So you have sympathetic pre, sympathetic post, parasympathetic pre, parasympathetic post. So parasympathetic pre and parasympathetic post are both, I'll type it out so you guys can see it. Parasympathetic pre is ACH. Oh, hold on. That is not too. Parasympathetic postganglionic neuron is also ACH. The sympathetic pre, again, is ACH because it's super important. Sorry, that was pre. And sympathetic post is norepinephrine. Hold on. Yeah, so the question was asking which is going to be prevented from being released. So when an action potential fires on a neuron, that neuron, when it makes it to the synaptic terminal, if the action potential makes it there, then it's going to be released. So the norepinephrine, if the action potential made it to the synaptic terminal, the norepinephrine would be released. But because it's not making it to the synaptic terminal, it's preventing norepinephrine from being released. Everybody understand that? Does that make sense to you, Anna? Cool. Okay, and I am so glad that we made it through that. Okay. So that's all the content that I have for today. I appreciate you guys sticking around and toughing it out. Do you guys have any questions before we go? So how can we get access to the slides? There's a OneDrive that we can share. Samantha, if you're part of the GroupMe, we'll put the documents in there. You can access all of them. We have Zoom recordings as well, and you can get them on YouTube as well. So all the information should be there. It's on our websites, and it's on our Instagram as well. Yeah, no problem. Happy to help. So that's the end of the lecture. You guys, if you want to stick around and ask a question or two, you're good. If not, you're free to go. Our next lecture is going to be tomorrow night at 8:30. And then we are still having office hours, I believe, on Sunday, July 4th at 8:30 as well. So if you have any questions regarding any of the lectures that we did this week, you can definitely bring them to office hours. Mark is going to be on, I should be on, a couple of the other tutors, and we'll be more than happy to help.
Hi, Chris. I did need to share my screen because there's this one nurse equation that they asked with the sodium potassium pump you mentioned. Do you mind I share it so you could take a look at the?
Yeah, you want to share your screen is what you're saying?
Yes, there's a question. Let's see, it is this one. All right. Are you seeing my screen? Look at question number 9.
Yes.
I asked, because I know you said it's not mentioned much, nurse.
Right.
But there are a few. So 9 was an issue for me. And let me just get rid of this. And if I scroll down, and they didn't give us the answers because they want us to think about it ourselves. So I really don't have the answer. So 9, I did the, the others made sense now that definitely number 9, and I think 4 with all those exchanges going on. The potassium 4 and 9. If you go over,
Wow. Yeah. So number 4 is talking about conductance and electrochemistry. That is probably a question that Mark is better suited to answer, if I'm completely honest. But scroll back up to 9. Let me see if I can take a look.
So which one?
To number 9. You said you were confused.
Yes.
All right. So a simplified version of the Nernst equation is shown below. What is the resting membrane potential of calcium? Calcium out is 1, and calcium 2.
I'm sorry.
That's okay. Is 10 to the negative 4th. So I think, are we just plugging it in? Did you plug the values into the Nernst equation? So
is that what I have to do?
Yeah, I'm pretty sure that that's.
Oh, okay.
There might be another step.
Because the log kind of scared me.
Yeah, logs can be a little bit intimidating. But
so all I have to do is substitute those values in.
Yeah, I believe that's what it is. It's just asking for the ion concentration in, which they give you. You put it on top and you put it under. You
I
take
can
the
do
log,
that and then take the log off. Yeah, I can do that. I was just wondering, how to go about.
Right.
So
whenever I'm confused about how to approach a question, I always look at the units. I always think that's such a great starting point. So, I mean, I'm just looking at the units, like we're given millimolar. So if you put millimolar over millimolar, it cancels out. And then we're left with that millivolts by that negative 60. And all of our answers are in millivolts. So that makes sense to me. But will they give you an answer key at any point?
Did they give us a what?
Will they give you an answer key at any point?
No, that's what they call it. Yeah, no, they said they want us to think. If I do, I'd have to send it to the Berkeley Review people for me the answer. You just submit it and then request. They don't just give it to you because they said they want you to think about it. So these are difficult ones.
Okay. Yeah, they are a little bit challenging.
So, okay, if you tell me to just go ahead and put these values in and see what the log of, and then I can complete this little thing.
Yeah, pretty much. And you can definitely use questions.
When you went over the PNS in number 10, did you go into details about Schwann and epidemial?
So I didn't mention astrocytes or ependymal cells. They are also glial cells. You can definitely look them up. They're just quick definitions. The ependymal cells, I know astrocytes have to do with, I believe they are responsible for the blood-brain barrier. Ependymal cells, they're slipping my mind, but a quick Google search will definitely clarify for you. Oligodendrocytes produce myelin in the central nervous system, and Schwann cells produce myelin in the peripheral nervous system. So it's the same exact function. It's just different parts of the nervous system.
Are these high-yield questions? Because I don't know if I want to spend.
Are they what kind of questions? What did you say?
High yield, meaning are they well-tested questions, or are they just trying to
yeah, so like number 10, I would definitely know. I would definitely just like review any sort of question like that where a quick Google search would clarify just like basic definitions, I think is definitely worth your time because those are the easiest to remember in. But something like the Nernst equation, honestly, maybe at most you'll see 1 question or 2 questions on the MCAT. So I personally think that as long as you're familiar with it, that's enough knowledge.
How do you do that without a calculator? The log without a I don't even know how to solve that. I'm looking at it and I'm like, okay, well, this is what it is plugged in, but how do you solve that? They're expecting you to do logs without a calculator.
Yeah, so you have to kind of do some manipulation. That's why they don't really use the Nernst equation a lot. You have to kind of.
But even with pH problems, they use it with logs. So I mean, you're not really getting out of it. But so I just want to know, how do you do the math with?
You kind of have to move some. You kind of have to move some variables around. So that log 10, the 10 is your base. So it's basically saying, if we move, it might be easier. Like if you move the negative 60 to the other side of the equal sign, you get negative E over 60. So it's saying 10 to the E over 60 is equal to that ion concentration. I know that kind of sounds complex when I say it out loud, but if you want like a visual aid, you can definitely find it on Google. They will talk about exponents and logs in the same lesson probably, and that's basically what it is. A log is basically just the opposite of an exponent. So it's basically just saying 10 to what power will get you the ion in over ion out. So that's how you would solve it. You would just do like basic math.
Yeah, Keith. So log is definitely something that comes up a lot throughout the MCAT for other calculations, but there's also a bunch of other, I guess, like basic math principles that also the MCAT requires you guys to know. And I know when I was reviewing the Kaplan review books, they actually have a section that's all dedicated to like the math that you would need to know. And so that's when they go over logs, like natural logs, common squares and things like that. Even like some of the signed values. So I think it would be really useful to go over that, to know like what kind of math they expect you to know.
I'm sure there are a lot of free resources online that you can look that will give you a guide to what kind of math that you need. Because any math that you have to do, no matter how complicated it seems on the MCAT, it's possible to do without a calculator. They would not put it on the MCAT if it were not possible without a calculator.
And my last question, Chris, because all of these you just went over, so I'm going to do the exam right now, right after, since I've had the lecture. Number 6, you did not go over nicotinic.
Right. No, I didn't go over nicotinic.
Talk about agonist and acid ACH, but none of this was mentioned in today's lecture.
Right. Yeah, so I didn't go over that just because of the time crunch. I would familiarize yourself with it though. Like the alpha adrenergic, beta adrenergic receptors, I would know the difference between them, at least in concept. Because I mean, like I remember learning about them in college and we did these huge lectures, but I would just like know at least just like the difference between the 4 of those and just like their main responsibility. So, I mean, the MCAT, like your conceptual knowledge will carry you a very long way with the MCAT. So sometimes just like for time's sake, we have to, you know, sacrifice concepts and just retain just like contextual knowledge about it.
So Janelle, remember I was having trouble with antagonist and agonist. So this question is telling me it's an inhibitor. Like I'm going to go ahead and Google what these different receptors are. So Janelle, when it's telling me it's an antagonist, it's telling me it's blocking the ACH, correct? Because it's then it's an agonist. Like
well,
it's an antagonist and then it's an agonist. What is going on?
Well, this one is like 2 statements. So it's saying Curer is an antagonist, and then aside from that, acetylcholine is an agonist to what? So like they're 2 separate. So they're saying one is one of these is an antagonist, and then the other one is an agonist.
Really asking about the ACH, not the curare. Okay, I get it now. You're just like curare is an antagonist. Acetylcholine is an agonist to what receptor? Okay, I think I understand it. Okay, you've covered everything on this thing, so I'm going to go ahead and do the test and request that. I'm going to stop sharing.
Yeah.
But thank you so much.
No problem. Best of luck, and bring any questions you have to office hours.
Certainly, I'm going to, like I said, I'll do the exam now, submit it, and have them send me the answer.
Awesome. All right. Any other questions?
Yeah, Chris. Is it important to know? Because I think I would think I was okay up until like when we first got to nervous system, just because I thought there was a lot of like different terminology just thrown around.
Yeah.
Is it important to know all these different ganglions and all this stuff about like the nervous system? Because I really am confused on like what to know, because I feel like this can get really into the weeds of very specific nitpicky stuff, and I don't know if like, is it worth really to know all of this, or is it just to try to get a big picture of it? Because
I think it's pretty worth it to know it, honestly. I know that's not the answer you want to hear, but I think the nervous system is pretty high yield on the MCAT. And understanding this will only help you in the other sections. Like Janelle said before, it pops up in the psych and soc, that section as well. So I do think it's pretty important. I would just recommend like taking the PowerPoint, going through it and maybe translating the notes into your own words, try to break it down visually, use charts, anything that can help you process the information better, and use your online resources. Watch videos about it, because there's a chance that someone else is going to explain it in a better way that you'll understand than I am. But I do think that a lot of the information on this PowerPoint, you really should try to commit to memory. And then some.
That's not good.
Yeah.
That is not good. This is gonna take me forever
it's gonna take some time.
Really to get down. Like I'm over it.
Right. It's not impossible though. It's gonna take time. It's gonna take effort, but it's not impossible. It is possible. You just have to work diligently. Okay. All right. Does anybody else have any parting thoughts? All right, guys, then keep a lookout. We will upload the PowerPoint, we'll upload the video, and hopefully we will see you tomorrow at 8:30. All right, have a good one, everyone.